Good morning. I wanted to welcome you all to the first ever industry session developed by the Industry and Institution Advisory Committee. The mission of this committee is to bridge the gap between APSA and industry in a manner that supports the objectives of our organization. This session was envisioned as a way to facilitate interaction in a mutually beneficial way between APSA membership and industry members, with the hope that the membership can gain insight into the cutting-edge treatments and current therapies on the horizon in pediatric surgery, and industry sponsors can engage with members in a way that advises and engages people interested in these projects. So I'm pleased to introduce the session, Innovations in Hope, Emerging Technologies in Tissue Engineering and Regenerative Medicine, sponsored by Biostage. Our first speaker is Dr. Joseph Vacanti, who is speaking on tissue engineering, regenerating hope for the future. Dr. Vacanti.
Thank you, Karen. Well, good morning, everyone. It's a pleasure to be here to introduce this session, which is novel. And before I start, I first would like to extend our profound thanks to Biostage, because they have been extraordinarily generous in supporting this whole meeting. And I think having this session to start off a new discussion about how to interact with industry is really good. Now, I am one of the co-chairs of the Scientific Advisory Board of Biostage, and it is really, it's opened my eyes for many, many things. But what I'd like to do is just very briefly sort of put an overview on how excited I am that this session exists today, because our first paper in this field that's now called tissue engineering, the very first paper was 32 years ago, and it was presented here at APSA. And even though it was science, I thought it very important that the concept be introduced with my peers, clinicians who really wanted to do everything we knew to do to advance the care of children. So our first paper was here, and now we can look 32 years later and see what progress has been made in the field and where the field stands relative to actually impacting the care of children and adults. So this is very meaningful to me. Now, I made a sort of an editorial comment a few days ago at that extraordinary session that Diana Farmer hosted with all the very senior members of the organization sitting up front and really discussing the history. And my comment related to the grace and the focus that it took for all of those people to actually move the ball in terms of creating the specialty, helping the children, and really what it took to actually advance that. And the same is true for this field of tissue engineering. One can look and be very pleased and proud, but also one knows, and I've lived some of that, so I'm only going to tell two quick stories as it relates to that the road, all of you who expect to try to advance the field in whatever area you choose, there are bumps in the road. Some of those bumps can knock you off a cliff, but those that sort of can pick themselves up and keep persisting is a very important trait. So when we wrote the first abstract to present our work after 2 years to APSA, my peers accepted the abstract. So it was a 10-minute oral presentation. I was very pleased, but I was fairly young. So I circulated my manuscript to some of my senior faculty members who I respected. And one of those people approached me before the meeting, probably a day before the presentation, and trying to be kind, suggested that I withdraw the paper. And so I wondered why, and I ultimately said, well, my peers have accepted this. I I understand you don't want me to embarrass myself, but I'm going to go ahead and whatever is. So that was the start. And then a member of the publications committee at the end of the meeting approached me and said, we're sorry, we're rejecting your paper. It wasn't returned with revisions, it was, we're rejecting your paper. So what do you do with that? And at the end of the day, we ended up modifying the paper substantially, and then it was published. 2 to 3 years later, as we were preparing another paper, in those days, there was no PubMed. You went to the library. You went to the Index Medicus to do the references, the list of references, what's happened in the field, and so that you can be appropriately, people understand where things stand relative to the work. And as I pulled those papers, some of the words looked really familiar. And as I pulled more of the papers, there were other phrases and words and paragraphs that looked really familiar. So as it turns out, the opposite had occurred within 2 to 3 years, where not only was there data that we had generated that was being published by others, but our actual words and phrases were being published. It was out and out plagiarism. So I'm telling those stories only for the young people to understand that even though you can see linear progress over time, those of you who choose to do this need to understand from those of us who've lived this that there's bumps in the road, and you need to rely on your senior people to help you through those bumps. So with that as an overarching sort of welcome and warning and encouragement, I'd like to introduce Bill Fodor as the first speaker, who's the Chief Scientific Officer of Biostage, and their work toward the creation of an esophagus for hopefully for the repair of esophageal atresia. Thank you.
Thank you, Dr. Vacanti. It's a pleasure to be here. What I'll describe for you today is our lead candidate product at Biostage. Biostage is a small biotech company located in the greater Boston area with a focus on tissue-engineered combination products for tissue repair. Let me see if I can... So our lead product candidate is a cell-span esophageal implant. It is a combination product. It's both a biologic as well as a cell delivery device. It delivers directly adipose-derived mesenchymal stem cells to the esophagus on a modified polyurethane cell frame scaffold. The cell frame scaffold provides temporary support during initial tissue ingrowth and then is removed endoscopically at 21 days post-implant. So it's a temporary device. Cells are then retained within that host tissue. Host tissue is stimulated to form an adventitial layer around that, and I'll show you some histological evidence of not only adventitial regeneration but also mucosal regeneration. The intended uses are for esophageal reconstruction for end-stage esophageal disease as well as pediatric atresia. We think that the product will have its greatest impact in pediatric atresia. The mechanism of action, and I'll show you data around this, was to stimulate host ingrowth, stimulate a constructive wound healing process, including extracellular matrix deposition as well as angiogenesis. This is the cell frame delivery device. It's an electrospun polyurethane. We can make it of any size dimensions. This is just SEM looking at the fibers. We characterize these by fiber diameter, pore size, tensile strength, and we do a surface plasma treatment prior to gamma sterilization, which allows for wettability of the scaffold and promotes cell adhesion. The drug substance, in this case, is adipose-derived MSCs. These are autologous, so there's no concern about the immune system. We actually expand them in vitro in 2D cell culture format following isolation from a fat biopsy. We characterize these primarily by ISCT standards, looking at expression of CD73, 105, and CD90. We also have done a lot of work around the mechanism of action. The cells secrete cytokines and growth factors. They lay down extracellular matrix, which has been characterized through proteomic analysis. And we know they stimulate angiogenesis, and we use an in vitro vasculogenesis assay, which when you take conditioned media from the ad MSCs that are grown in culture and you expose human umbilical vein endothelial cells to that conditioned media, they actually form capillary structures in vitro. So it's a functional assay to pinpoint the mechanism of action. This is the product overview and our specs. Again, expanding the cells in 2D culture. We have a custom-made bioreactor, fully closed system now, automatic seeding. We take the cells and seed the scaffold, incubate that in the custom bioreactor for 6 days. The esophageal implant is then harvested from that bioreactor. This is just an SEM to show you that the green cells all intercalate in between all the fibers, which are indicated by the red. Following that, during that 6-day incubation period, we do full characterization of the metabolic profile. At the time of harvest, we have a QC segment where we do all our QC release assays. This is just a product paradigm. Again, producing the scaffold, adipose tissue biopsy, characterizing the cells, putting them into the bioreactor, and then for surgical implantation, we do a segmental reconstruction of the esophagus by taking out 5 centimeters of the esophagus, and this is done in the Yucatan mini pig model. And then we implant the cell span esophageal implant. With respect to the animal model, we've done a series of large animal studies in older animals as well as younger animals. In fact, Dr. Christine Fink presented on Sunday some of the work that we're doing with her and her group at Connecticut Children's in a pediatric model of esophageal disease. At the time of implant, the scaffold remains for the first 21 days. It's then removed at day 21, and what we're left with is a complete tube of tissue. We actually put stents in there to hold the tissue open during the healing process, and those can go out to approximately 6 months. At the end of the study, we do the histology and the mechanical testing. Again, our target indications are for end-stage esophageal disease as well as pediatric atresia. So in order to understand the regeneration process, we did a whole series of proof of concept as well as GLP studies. This is an acute tox and biodistribution study where we used GFP-labeled cells, and we did a time course here. We had animals that were harvested at 2 weeks, 3 weeks, and 4 weeks, and what you can see here is you have a complete tube of tissue. This is the esophageal region of the implant where it's been filleted open so we can look at the luminal surface. We start to see the migration of epithelial cells into that center of the implant zone over the time course, and that implant zone becomes epithelialized completely by 3 months. But again, this short time course, we get fibrovascular tissue in the implant zone, and we know that those cells actually contribute to new vascular structures, and you can see the green cells here surrounding a capillary structure within the implant region. Longer-term studies, again, a whole time course of how we believe the regeneration process is occurring. Again, 30 days, we have this unepithelialized region. By 90 days, it's complete. By 9 months, we have a complete epithelium, and we start to see some smooth muscle cell migration into that implant zone. And by 600 days, we had 2 animals go out 1.7 years. We have a complete epithelium. We have a near-complete muscularis mucosa. We have smooth muscle cell, and you can see a lot of smooth muscle cell actually replacing all that fibrovascular tissue. So we believe that the regeneration process takes quite a bit of time, but what we are seeing is almost a recapitulation of what the native tissue looks like. And again, formation of the epithelial cell layer, the muscularis mucosa, and now the tunica muscularis development. What happens in humans? We are fortunate enough to have a collaboration with Dennis Weigel at the Mayo Clinic. He had a patient that required esophageal reconstruction. The patient also required vena cava reconstruction as well as some heart patches. This was a 75-year-old male. We enrolled him into an emergency EIND compassionate use application. It got approved by the FDA, and Dennis implanted the Biostage CEI into this patient. He went through quite a bit of surgical reconstruction with the heart, the vena cava, as well as the esophagus. Just to give you a preliminary patient histology, we were able to look at the tissue post-mortem. The patient actually died of a stroke 7.5 months after the surgical implantation. We compared that to what we see in the pig, and we looked at native esophagus, the transition zone, as well as the implant zone. And again, we see very similar regeneration within the human as compared to the pig. And most importantly, we get regeneration and recapitulation of a multilaminate tissue. So we have the epithelium, the lamina propria, and the muscularis mucosae. And again, as time goes, we see smooth muscle cell migration into that implant region. This is just a summary of how we believe that the CEI implant works. Again, we implant this by making an end-to-end anastomosis to the native esophagus. The scaffold remains for 3 weeks. The MSCs are seated onto that scaffold. We have ingrowth of host tissue into this fibrovascular zone. At 3 weeks, the scaffold is removed. We're left with that fibrovascular tissue and again, a complete biological tube. Over time, we get smooth muscle cell migration. The adventitia develops, and we get complete epithelial mucosal regeneration. In summary, we have demonstrated both through POC as well as GLP preclinical studies with new tissue ingrowth. We have luminal epithelial migration and mucosal regeneration within the first 3 months. We have stimulation of angiogenesis within the first 2 weeks, and so we never see a necrotic area of tissue in the implant. It's a functional biological conduit, and we've had survival up to 600 days. We completed that EIND compassionate use case. At 7.5 months, we were able to take a look at the tissue and observe the regeneration process occurs very comparable to what we see in the animals. It is our platform technology and can be tailored for other hollow organs and tubular structures. Just want to acknowledge all our collaborators. Dennis at the Mayo Clinic, UT, and Chuck Cox's group, our collaboration with Connecticut Children's, Boston Biomedical Associates as our regulatory consultants, and all our animal CROs. Thank you.
Thank you. So we're going to move on.
Should I introduce Chris?
Yes, please.
Chris, come on up, please.
All right. This is Christopher Brewer. He's from Nationwide Children's, going to speak on tissue-engineered vascular grafts. Thank you.
Well, good morning, and I too would like to thank Biostage for their generous support of APSA and of this section. For the past 25 years, I've worked with my collaborator, Dr. Toshi Shinoka, and focused on trying to apply tissue engineering to children with congenital heart disease that require reconstructive operations. And the impetus for our work is that most of these major reconstructive operations require the use of man-made materials, which are a significant source of morbidity and mortality. And how do we do it? Well, we start with a biodegradable tube that's called a scaffold, and this tube is manufactured from the same material that absorbable sutures are made from. And then onto this tube, we seed an individual's own cells. Specifically, we seed a fraction of the bone marrow called bone marrow-derived mononuclear cells that are seeded into the porous wall of the scaffold using a vacuum seeding method that uses negative pressure to draw the cells into the wall. And then the scaffold actually serves as a template that directs tissue formation. Now, once seeded, the scaffold is ready for immediate implantation as a vascular graft. And once implanted, the scaffold induces a foreign body reaction in which the host cells actually infiltrate the scaffold, specifically monocytes, which transform into macrophages. And as this foreign body reaction is occurring, the seeded cells are actually disappearing. Now, over the last 10 years, we've discovered that these macrophages are actually very important for the process of tissue formation, and what they actually do is they induce the ingrowth of the native smooth muscle cells and endothelial cells along the luminal surface of the scaffold. And as this vascular neotissue forms, the scaffolding degrades and the inflammatory cells go away. And what you're left with after about 6 months is a living neovessel that has the ability to repair, remodel, and even grow with the host. So our initial clinical target has been the use of the tissue-engineered vascular graft for use in the Fontan operation, where a vascular graft is needed to connect the inferior vena cava to the pulmonary artery. We selected this initial clinical target not because of the great need for a vascular graft in this space, but because of its excellent safety profile and because these patients would have the potential to benefit from the growth capacity of the tissue-engineered vascular graft. And we performed the first implantation of a tissue-engineered vascular graft in the United States in this little girl named Angela. She completed the study over 7 years ago. Serial MRI imaging of her graft demonstrated that as she's growing, so too has her vascular graft, making the tissue-engineered vascular graft the first man-made vascular graft with growth capacity. We've had several other children in the study. This is Anderson. Anderson developed a narrowing of his graft, which was seen in several of our patients. This stenosis was managed using angioplasty. All of our patients in this trial developed stenosis early on, within the first 6 months after implantation, and all are alive and well with grafts that are growing now a minimum of 5 years after implantation. But because of the high incidence of early stenosis, we went back to the bench and tried to understand what we call the process of vascular neotissue formation, or how does a biodegradable tube seeded with bone marrow cells transform into a living blood vessel. And one of the things that has proved to be very useful over the last 5 years has been the use of computational modeling. We've actually been able to develop a model that can actually describe and predict neotissue formation in our tissue-engineered vascular grafts. And this might seem fantastic or maybe surreal, but it really comes from the fact that there are two processes that control the neotissue formation, and these processes are quantifiable and can be correlated with outcomes and are therefore predictable and amenable to modeling. One of the things that we've shown is that the primary driver of neotissue formation is inflammation. The host inflammatory cells infiltrate the scaffold and begin to release cytokines, which through a paracrine mechanism, induce the ingrowth of the native cells. Now, the degree of inflammation and the amount of cytokines released is controlled by the scaffold. So by controlling the chemical composition and morphology of the scaffolds, you can control this aspect of neotissue formation. More recently, we've really begun to focus on the mechanobiologic factors that control tissue formation. And these prove to be extremely important in the remodeling processes that occur a little bit later. And to understand this, you need to understand a basic concept that's called mechanical homeostasis. And that refers to the fact that the smooth muscle cells that make up the wall of the neovessels have a predetermined or are hardwired to have a predetermined amount of mechanical stress that they need to experience in order to be quiescent. And if that is perturbed, then they react in a predictable fashion. So if a smooth muscle cell experiences too much stress, it will actually begin to form extracellular matrix, creating a cocoon around itself, or stress shielding, and it will continue to produce extracellular matrix until that set point, that mechanical homeostasis, is reachieved. Similarly, if a cell is understimulated, it will break down the extracellular matrix until it experiences that predetermined set point. So if you can measure the mechanical components of your graft, you can predict its behavior. Now, interestingly, we started to use this model and perform simulations. And over time, multiple simulations replicated what we saw in our clinical trial. Our vascular grafts would narrow immediately after implantation. But what the model also predicted was quite surprising, that this narrowing would go away by itself without any intervention. So this gave us a marvelous opportunity to try and validate or test this model. And so we performed a series of large animal implants where we would replace the inferior vena cava in an ovine model. And what we saw is that 1 week after implantation, we had a widely patent graft. By 6 weeks after implantation, virtually every animal had a tight stenosis that would have met indications for undergoing angioplasty in our clinical trial. But because we wanted to test and validate our model, we didn't perform angioplasty and simply followed these animals. And by 6 months, the stenosis had resolved spontaneously. And between 6 months and a year, the grafts began to grow with the animals again. Similarly, during this time, over 90% of these animals were completely asymptomatic, and the stenosis wouldn't have been detected if we weren't carefully monitoring these animals. So taken together, these findings suggest that we overtreated our patients in the clinical trial, and that this early stenosis is actually a part of the natural history of new tissue formation and can be safely observed in the majority of patients. Now, the model explains it a little bit further. It suggests that we have an early period of inflammation that induces excessive tissue formation, which creates a very non-compliant graft. Over time, as that scaffolding degrades, the vessel becomes more compliant, and we get some of that mechano-mediated remodeling, which ultimately results in the widely patent neovessel. So moving forward, we've shared these data with the FDA and are beginning the next phase of our clinical trial in September of this year, where we'll look at the tissue-engineered vascular grafts. And in addition, we've changed our indications for angioplasty based on our computational modeling studies, in addition to adding some additional strategies that we've discovered over the years can also be used to inhibit the formation of stenosis. Thank you.
Thank you very much. Our next speaker is Seppi Hashemi. She's the Vice President of Program Management at Boston Biomedical Associates, and she's going to speak on product development, pathway, and clinical translation.
It is a pleasure speaking to you, and it's a privilege to be a part of the Biostage team for the past many months. Boston Biomedical is a specialized device, clinical, regulatory, and product development organization in Marlborough, and we are really integral to the Biostage team. My hope here is to very briefly follow in the very thoughtful and provocative presentation by Bill and Chris, and maybe share some ideas as to how industry views collaboration, bridging with the academic teams, as well as my experience at Partners Healthcare Innovation, where I actually was fortunate enough to be sitting with many of you in the worlds that you operate in and try to make that connection happen. And then we're going to just move to discussion. Let's see. Okay. There we go. So very briefly, as someone who's lived in innovation land, I wanted to maybe offer up 3 thoughts here as to how it actually happens, or the way you think about it. The first is that it truly is guided by disease insights and technology breakthrough. You just heard Chris demonstrate that exceptionally well. The role you play in identifying the disease breakthroughs is unparalleled in industry, even for folks who transition from academia to industry. That is something that's quite rare and quite special, and that's why industry would come to see you always. Secondarily is that the technology breakthroughs used to sometimes be viewed as just the companies bringing it forth, but clearly, as time has evolved, and as Dr. Vacanti pointed out, it's also from academic teams observing the value of technology. One of the great examples is use of biomaterials, especially in the space that you all operate in. The second is that the way the world of commercialization views it, innovation is an invention until it actually becomes used and generates value. That's when it really becomes an innovation. And the third is that it is almost always catalyzed by hybrid teams who are vertically integrated and have hybrid skills, and they can look through the lens of both the technology, the disease, and how a product is produced. This idea of looking through stakeholders' lens is an important way for you to think about it, particularly those of you in the audience who are early career as you meet with industry, is to really look at it the way you are very comfortably looking at it from a patient's view. The device side almost always is a reduction of symptoms and risk view for the patient. Clearly, the regulator is needing to be convinced around the uncertainties. Many of you as early adopters are very clear on how you want to solve a true perceived unmet need. It is the clinical user, a broader user of product in a larger or smaller clinical setting, that often helps industry with identifying what treatment options and what populations, definition of populations and sites help generate the clinical data that moves the program forward. speaking on behalf of at least one large medical device company, I could tell you these are actually words of a CEO of a device company asking this question of all of the engineers who would show up in his Friday review meetings. Does it work? Can we prove it? Who really believes us? Can we manufacture this thing profitably? And that is a different thing with industry, right? So if you touch industry, you're going to hear the words of value and profitability. And who will pay for it is a new and important aspect here. This is beyond regulation. This is reimbursement. This is CMS asking what is payable, if you will. Obviously, investors are purely looking at return on a desired window of time. And lastly, and as I mentioned, payers who are really looking at natural history and truly long-term outcomes. And I think for the pediatric space, this is a very important area to keep in mind. So one of the things I would ask for all of you to think about as a paradigm here is typically coming from an engineering background, there is a tendency to think linearly about I reduce my uncertainties at the bench and in vitro level. I use animal studies to have a window into the clinical performance, and then I consider risk and benefit balance with my esteemed academic partners. I would tell you, in all cases, insights are inferred where you start with the first-in-human study first. Really, really think about what the indication for use is, what are you validating in a very small clinical study, and what features and procedures need to be refined, particularly if it's a surgical product. The opportunity to reduce surgical risk is profound in the space that you operate in. It is important, and many folks in this audience are highly skilled to think about clinical strategy as that overall roadmap of evidence to support product performance. All of those things you see there are highly valued by companies. All of them tend to reduce uncertainty, enable critical decisions, and really give you a sense of when is it likely to generate some human data. And for industry, that human data is really the driving factor for decision to fund a program, continue to collaborate, extend research projects, etc. So again, expectations of first in human, many of you are quite familiar with it, but it is important to think about it as a why, how, what. Should the patient accept the risk? Is the unmet need real? In your space, by spades. I mean, it's a space I've chased for many years from device side, because if there is a place you want to make a difference, this is it. It's not just about how does the device work. This is a bummer of a conversation for industry when you tell them how does it fail. You have to think about how it's going to fail or how it needs to be maintained if it's a growing device as it is in the pediatric space. And then lastly, most importantly, what decisions are enabled by the data that comes from a first-in-human use program? Are you enabling product design, looking at indications, really honing in on profile of the patient? Are you looking for user skill development? I think we've talked about patient-centered risk-benefit. This is an audience that's quite familiar with it, so I'm not going to spend a lot of time on this, but it is one of the most valuable ways all of you calibrate industry. I encourage you to speak about this very often. It causes convergence. I do want to point out that the agency has come a long way in establishing an early feasibility study process for devices. I want to encourage you to read about this guidance document. There are some really good, important nuggets in there. It tends to drive programs. It's an acknowledgement that early feasibility studies can be done in a setting where the entire product is not verified and validated, but it's in a controlled way where the intended clinical use is very novel. The design of the study is anticipating that there might be product or procedural refinements, and the test methods are limited, which many times in the pediatric case there might be. The viability of the animal models or limitations of non-clinical allows you to think about risk-benefit and move towards an early feasibility study. The other item I want to make sure that you guys think about as industry folks think about it is this notion of design control. And how do you know that the product is actually ready to go into a person anyway? And if I could leave you with one thing, it's simply this idea that there's an understanding what design input is, meaning what are the specifications or the expected user performance for a product. and then the design output, which is what is the objective evidence collected. If you hit this design input equals design output robustly, I promise you, you will have convergence more often with the agency, because their challenge often, as being risk averse in wanting to reduce uncertainty, is whether that non-clinical or clinical evaluation method is predictive. And I think what Chris just described in his method for theoretical analysis is a shining example of what would be smiled upon. So this EFS type approach is really about looking at device mechanism of action, really honing in on patient population. Those of you who have been part of clinical study programs with the industry know that it will take hours and hours to get to the clinical synopsis. But if you get to a very good one, it paves the way for a very good conversation with the agency. The idea of patient characteristics impacting device performance is critical. In a surgical space, being able to really think about the technique challenges and get to as flawless a procedure in a first human use model is really critical. Obviously, the focus is on device safety. And if you go through this process, I think the more you think about how it would be studied, the more it helps you think about the non-clinical testing that you need to do, or streamlining it so it's not blown up into something that's not even achievable in a hybrid team between academic and the industry team. So just to wrap up, how does this apply to pediatric innovation? You are operating in a space where the design input side of that design control I mentioned to you has a very unusual flavor to it, because the recipient is growing. That is much, much more difficult than when I worked on stents and vena cava filters. You put it there, nothing much moved. Maybe you had some plaque, or maybe you had to go touch it up. You're talking about dimensions changing. So it does require a different type of looking at it and positioning it for folks to wrap around it. And I think one of the things, the beauty part of tissue engineering is there really isn't anything polymeric on its own without cells that would work in a growing model anyway. There never would have been. Even if everybody was excited about Dacron or PTFE, on its own, it's never going to fly. So I see Dr. Bacanti smiling, because it's just a validation of what he's been looking at for all these many years of elegant work. Animal model complexity is part of this. Procedure variation versus adult is obviously part of it. One thing that's been paid great attention to by the Biostage team is the notion of inclusion-exclusion as they begin to concentrate on a first-in-human program. Critical, whether you're looking at an adult indication or a child. And then lastly, as you look towards clinical operations and execution of a trial, the consent is really through a parent, not a patient. And I think you're all very well aware of it. Is industry always very well aware of that? Not necessarily. You are the educators of how people think about risk-benefit through the lens of the patient and through the lens of the parent as a surrogate. So with that, I appreciate this opportunity to speak with you. It's always a pleasure to be in the clinical audience, and I look forward to the discussion.
Thank you very much. Bill, why don't you come on up, and then we'll take some questions from the audience. Hello.
Hi, Marshall Schwartz from Winston-Salem. A couple questions regarding the scaffold, Bill, that you mentioned initially, that you remove it. It's a polymer, is that correct?
Correct, it's polyurethane.
So with tissue ingrowth, with cell ingrowth, tissue ingrowth, I would think that would be difficult to remove.
No, at 21 days, the cells don't like it anymore. To put it simply, that plasma coating that we put on to make the scaffold hydrophilic allows for cell adhesion. We think by 21 days the quote-unquote plasma coating has been consumed and the cells no longer have a preference to adhere to it. And so it just comes out on its own. In fact, in some of the first experiments prior to my time at Biostage, the scaffold was actually found in the stomach. So it actually...
Is the scaffold by itself, let's say if it stayed there, is it biodegradable or is it something that doesn't biodegrade?
It's not biodegradable.
Okay.
It's polyurethane. It's
Okay.
plastic.
Okay, thank you.
Next, we have a few more minutes for questions.
Quick comment, Peter Liebert from New York. I was at the meeting when Jay Vicanti presented APSA with the first results of tissue engineering research. And I called my wife, who is a medical publisher, and I told her, this is an important area of surgical development. And so I just want to show everybody, and I'm leaving this with Jay, the latest copy of the journal Tissue Engineering. But I also do have a question. All of your histologic slides, which also show the ingrowth of smooth muscle, do not have a correlation with function. I imagine that 75-year-old man who had an implant had to have some peristalsis in that segment, and I wondered if any of that has been tested.
Well, we have not done the myometry to actually look at motility of that implant region. We do barium swallows to follow the flow of food, if you will, through the esophagus. We know we see, and it takes a while for it to develop, and typically what we see is really good motility above and below the implant zone, and we're still now looking at longer-term animals to determine how much motility is actually going through the implant zone. That's a very good question. But we do know that the implant region functions as a biological, conduit, and it allows food to pass through the esophagus.
Thank you.
In the blood vessels, they do become vasoreactive and constrict and dilate to pharmacologic reagents, and they basically overlap the constriction and dilation curves of the native vessel into which they're implanted. Takes a while, takes over a year for that to be fully functional.
But it's important.
Yes.
Yep.
One last question.
Paolo De Coppi, Great Ormond Street, London. I would like to thank all the speakers for the elegant presentations. A couple of questions, comment quickly to Biostage. Firstly, in our preclinical and also clinical experience of tissue engineering transplant, we have used the omentum to vascularize, in that case, the trachea. And are you considering on using a muscle flap or omentum to induce or help angiogenesis, or you think is not necessary? And the second question regards the cells, because you use adipose-derived, but we are, as pediatric surgeon, we actually counsel many patients with absent stomach and polyhydramnias, and so we could actually storage and use eventually the cord cells, for example, that could have a similar function, avoiding any morbidity derived adipose tissue from an infant. Thank you.
We have not done any experiments with the omentum or a muscle flap. We've considered, should we be seeding these scaffolds with not only MSCs but smooth muscle cells? When you start thinking about product development, it becomes very, very difficult to try to manufacture something like that. We believe that the regeneration process, although it takes a while to occur, we actually start to see recapitulation of the native tissue over time. And presumably that the peristalsis will also evolve during the regeneration process. We have just not had animals that have gone out that long. Thank you.
Thank you. I'd like to thank our speakers for their wonderful presentations and everyone's participation in our session. Thank you.